All right, everybody. So we are getting into DNA restriction enzymes and gel electrophoresis. But before we get there, I want to try and refresh your memory and think back to doing paper chromatography. So we ultimately smashed up a bunch of leaves and we found out that even though they all look green, there are different photosynthetic pigments within those leaves, and those different photosynthetic pigments were separated. And we could see that they all looked like just some mush of green, but once we separated them through paper chromatography, we realized that we could see different banding patterns here as the different molecules moved faster or slower through the paper. Now, if you had an unknown leaf and you smashed it up and compared it to a known leaf and they had the same RF values, you could say that they had the same, possibly the same photosynthetic pigments. So this is very actually similar to what we do with gel electrophoresis. So the big ideas here. DNA is a chemical. It can be broken down by enzymes or with the assistance of enzymes. Enzymes are specific to particular shapes. So certain patterns in the DNA are going to be targeted by certain enzymes because certain patterns can have certain shapes, and then you can have certain enzymes to find those patterns and then cut the DNA at those points. So the patterns of DNA code are different for different people. So much is identical between people. However, everyone is slightly different. We have same genes, but we have a lot of parts of our DNA that don't code for genes, and some of those parts are subject to mutations as well and can be different for different people. So if we look at DNA samples um, that from different people and they're all cut up, those bits of DNA can then be separated through gel electrophoresis the same way we did with paper chromatography. Similar ideas. The bits will be spread out as they are pulled through the gel. And we're going to take a look at what that all means here in a second. And if two samples of DNA from two different people, or from two different samples are pulled the same distances and those bits kind of line up, they're probably from the same person. So we're going to get into this. So basically, you find some DNA. Whenever you hear polymerase chain reaction or PCR, basically what it means is to make copies. Polymerase, making polymers, ACE, it's an enzyme. So you have this enzyme which, after you go through some of these processes of separating it, denaturing it, um, this enzyme can help reassemble, basically almost like going through DNA replication, and then you can get new strands. And then you can do this process, this chain reaction over and over and over and over and get just tons and tons and tons of copies of DNA. So even if you have a small sample, you can ultimately get a lot of DNA copies. So anytime you hear PCR or polymerase chain reaction, that's what they're talking about. So the next step is to chop it up. And there are another specific enzymes, restriction enzymes, and there can be different restriction enzymes to target different parts of the DNA. But these restriction enzymes are ultimately going to cut it when it finds particular patterns. This AATT, it's going to find that, it's going to snip it right there. And let's come back to paper chromatography for a second. We took the leaf, smashed it all up, added some solvent in order to basically take something that was one solid leaf and get those individual pigments on their own. So what we're doing here, when we have DNA, if we imagine this as like 4 samples of DNA here, this restriction enzyme, every time it finds this sequence, it's going to cut it. So let's say in this DNA strand, it found the sequence, it cut it. It found the sequence, it cut it. It found it, cut it, cut it, cut it. What you're left with after everything is cut is these different lengths or different bits of DNA. Now, for the next person, again, they're still human. We still have very similar DNA codes. However, maybe it finds this sequence here and it cuts it. And then it doesn't find it for a while and then it cuts it, but then it finds it right again and it cuts it again and it cuts it. And we have different lengths of bits of DNA code after this sequence is cut compared to this sequence. Same thing with the third sample here. Cuts, 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 different lengths. And when we run this through the gel, these would all look different. But then if we have another sample and we cut it and we cut it and we cut it and we cut it and we cut it, you'll see that these cuts are very similar to where these cuts are. So that kind of says maybe these DNA samples are from the same person, or at least closely related. So when we come back to our paper chromatography, we took our sample from our leaf and we end up running it through the paper as this capillary action works the solvent up the paper. And what we do with gel electrophoresis is we take some agar and we make it into a liquid and then we pour it into this little tray. Okay, so the tray, it's almost like a concrete form. And there's this little comb in there. And this comb, all this liquid agar goes all around all of these little combs on this thing here, and then it solidifies, gets hard, we pull the comb out, and now there's these little wells. There's these little spots where we can squirt stuff in. So that's basically what we do. And then we're going to take these samples and shoot them into the wells, turn on this electrode, and they're going to get pulled through. And again, thinking back to what we did with gel electrophoresis, or not gel electro, but paper chromatography, as those bits of pigments get pulled through the paper, because of their solubility and their affinity towards the paper, they're going to get pulled different distances. So again, over time, as they get drawn through the paper, they spread out, and then we can use that to actually do some data analysis and look at the R factors and stuff like that. So again, pretty similar when it comes to gel electrophoresis here. Yikes. Okay. Let's do this. Okay, so again, different little fragments here. Those little bits from each sample get shot into these wells, and then over time, the smaller bits are going to get pulled through faster and make it further away. When it's done running for a certain amount of time, we can take a look at the pattern and try and make some analysis. So let's look at something here. Crime scene analysis and paternity tests. So let's say you have a mom. She has a baby. Obviously the baby is hers, right? It came from her. But if we don't know who the dad is, we can use this same idea of restriction enzyme and gel electrophoresis to try and make some conclusions here. So you can see that mom and the infant are sharing some similarities here. See how they match up? Here and here and here and here. Now again, this baby is not a clone of mom, so it's not going to have the exact same DNA. But is the infant more similar to male 1 or male 2? Again, each of these are going to represent different bits of DNA that were cut, and the longer they go, the smaller they were. But we can make comparisons here. So if we take a look at male 1, that's there, that's there, that's there. So there's some that are the same, right? We take a look at male 2, this one matches up. So same with mom. This one, male 2 does not have, but mom has it. This one, Male 2 has, but male 1 does not, and neither does mom. So now we start thinking, hey, male 2's got something going on here. Again, male 2 is here, and this one goes with mom. This one does not go with male 1, however, it does go with male 2, and mom does not have it. This one goes with mom. This one goes here. So, if you're making an analysis here, male 2 would more likely be the dad. Let's take a look at some crime scenes. So all of a sudden we find some DNA at the crime scene and we run a gel on it. We also run a gel of the victim's DNA to see if this unknown DNA is the victim's DNA or if it could possibly relate to one of the suspects. And again, as we look through this, you're like, okay, this doesn't really match up that similar. But now, could it be suspect 1 or suspect 2? As you look here, this DNA sample is really suspect 1. And again, this can end up being a bit of a form of evidence to say like, all right, we got some DNA from this suspect at the crime scene. So we you're there at some point. So again, it's a way, this whole restriction enzymes, you cut apart the DNA, you run it through a gel to separate the different bits to try and make connections between, um, you can do it for paternity, you can do it for crime scene analysis, but it also gets into like looking for recessive disorders and different things like that because there are different restriction enzymes that target different points of the DNA. But ultimately that's it for this. So we are now finished with that.